Hi, I want to start by really thanking Sustainable Farming Fund. When we started our little pilot project, we didn't know if it would ever going to go anywhere. And we worked hard to get a Sustainable Farming Fund grant. We did. We got our first 3 years. And George Strachan is here. He's our rep, our SFF rep. I don't know where you are, George. Yeah, in the back. And SFF, they really help you get the grants. They're wonderful. And we've now have been approved pending some details that we may be able to do the next 3 years again, which is fantastic because when I show you what we've done so far, we're now on a roll and we can really get a lot of information on a lot of plants in the next 3 years. So there's a lot more authors than just me on this. I think it's only me on the program, but these are all the people on our team. Jean-Noel Galliot was our French intern up here. Lots of funny stories to tell about him. He's a fantastic intern. Finn Shield was our assistant in Landcare. Marco Gonzalez is in the head of the demonstration farms in Canterbury, and he's the apiculture officer from Assure Quality. Ian Rain is our palynologist. Karen Roger, our, I guess, geochemist or our isotope expert to do all the chemical analyses. Shun Lee, a palynologist. Gay Rattray, a pollen preparation expert at Landcare, and Tony Roper, an apiculture officer at Assure Quality. And it's a great, great team supported by John Hartnell, Barry Foster, and John McLean. So we're on a roll. Now, there's where we started, 2009, and those bee plant guides, I'm happy to say they stand because they are the plants that the beekeepers say are good, and we know they're good. So if you plant those, you know you're helping bees. And so if you figure out your flowering calendar, you're really helping bees, but we don't know how much protein is in them. So our whole goal of finding out protein is to be able to say, this is the superior maximum nutrition. So if you have a small space and you want to give them the best nutrition, then we want to know what protein is in the pollen. And so our goals are to increase that diversity. Barry has shown us, and Karen has shown us, and John has shown us that diversity is really important because you've got to balance out those amino acids. So also we want to maximize the protein in the pollen, and we want to fill in those pollen dearth gaps where the beehive goes backwards in its numbers. Because what happens in the beehive, I think has been mentioned, is that the nurse bees, if there's not enough pollen coming in, what they do is they start feeding the youngest babies to the older larvae to get the protein. They have to. It's triage. Yep, that's the do. And anyway, we're trying to avert a pollinator crisis. So we know, we've already seen all this, that it's very important. Our bees, it's our #1 important pollinator decline issue in New Zealand. The pollen dearth, why do we have pollen dearth? In the old days, you can read some of those old books on beekeeping and stuff, and there was quite a lot of pollen around. A lot of diversity of plants around on the farms, mixed farms. Now we've got an intensification of agriculture, so we've got more monoculture. We've got land use changes with dairy, losing land to dairy and urban, and dairy, the way it's done now, is just ryegrass, a lot of times, not all the time. Monocultures, that's just, there you've got that diversity issue. Weed-free farming, you got the herbicide spray and you just spray out all the weeds. And you don't want any weeds. And so that has a big effect on your agriculture ecosystem. And then, now this is important. In New Zealand, we've got a different problem that is really, not unique to New Zealand, but special to New Zealand in that it kind of messes us up. In New Zealand, we don't want the weeds, because the weeds are destroying the environment. We're an island nation like Hawaii, and this is serious business. We cannot have these weeds. Unfortunately, a lot of these weeds, which I'm going to show you, are some of the best bee forage going. So that's why we have trees for bees. Now we know a little bit about the protein values. There's things like pine only have 9% in them. And when my first beekeeper meeting I ever went to, John Syme stood up and said, "Can you tell me why my bees don't do well on pine pollen?" And I just started, I don't know. And now, about 6 years later, I'm saying, well, now I know why. Bees don't do well on pine pollen because it's very, very low in protein. And the other things, the willows are a little bit variable, but anyway, got lots of different, it ranges from about 7% to about 35% protein in pollen is the range that I've seen so far. So we're focusing on pollen dearth, and in this talk and in this conference, we're focusing on the spring buildup, and because we haven't got the winter stuff done yet, the autumn stuff. That's what I'm going to be doing right now. We're trying to find which plants have protein-rich pollen. We're trying to find, you remember that problem I had with the farmers? They don't want my list. We have to find multi-purpose plants that actually work on the farms. And we were asked in the beginning, much to my horror, to do a demonstration farm, because I'm not a farmer and I didn't know anything about it. Now I'm very glad we're doing it, because it's proving everything. So our first issue was, how are we going to collect pollen? Well, we've got 4 ways to do it. We can get it from traps on the hives, where you just, they scrape the pollen pellets off the bees when they go into the hive, and you get a bucket. That's when you buy pollen in the health food store, that's how they get it. Pollen loads at the hive entrance, you can catch the bees. I can't, beekeepers can, catch the bees at the entrance of the hive with pollen loads, or you can catch them in a flower, which is what I prefer, or you can directly extract pollen out of the flower. Of those 4 methods, there's pros and cons of each method, and we use all 4, depending upon all the circumstances of how hard it is. It's about labor intensiveness or even possibilities of sometimes you can't get pollen except through a hive. But our preference is to do this one where we're getting it on the flower. So the pollen loads, some people, experienced beekeepers, can kind of look at the color of a pollen. Can you see the arrows pointing towards the pollen load? Pollen load in the basket, that brown is clover, and red is horse chestnut. These are very distinctive. Poppy is gray. Brassica is yellow, but so are a lot of other pollen. Thyme is white, but so is a lot of other pollen. And rock rose is a bright orange, but so is a lot of other pollen. And so when we started, it was kind of like, do we know that this pollen from this bee on this leg came out of that flower? And when we had the pollen out of the traps, we said, can we sort the red, the yellow, and the blue, or whatever color, and say that we know, this is before I met Karen, and we were supposed to do 150 pollen pellets, for a nitrogen sample in an old-fashioned method. And we were pretty upset when we found out, no, they're different pollen in that color. And no, sometimes the pollen that comes out of the bee leg in the flower that you got it from is not that flower's pollen. It's the pollen from across the road. On another, that happened to us. So when I give you the results I'm giving you today, I want you to know, A, these results are preliminary because we have a way of checking everything, which I'm going to show you. And I'm going to give the results because the error rate will be only about 5%. But it is preliminary data, and in June and July, Ian Rain will be confirming and checking that our pollen is really that identity. And then we'll be publishing, before next November, we will be publishing our list with the protein values. So we take the little sample, and we have to take a little corner of it to make a pollen slide to get it verified that, yes, that's that pollen. And then we take the bigger part of the sample to send to Karen for nitrogen. Okay, so that's our process. And we do this acetylysis process, which means that we clear the pollen. We chemically clear all the contents out of the pollen grain, and then you get a clearer picture. That's broom, and this side here is this is the unacetylized. If you just plonk pollen onto a slide, that's what you'll get, and put a little stain, that's what you get. But if you put it through a clearing process, you get a clearer picture of the features. You need those features to identify your pollen. There's willow, and you can see willow looks really different when you acetylize it. So that's part of our process. And that acetylizing has to be done in a fume hood. You've been dealing with big acids, a lot of acids, and so it's, you have to do that on a fume hood. And then our step 2 is we go to Ian Rain and say, is that pollen the pollen that do those pollen match between what's on the bee leg and what's in the flower? Are we correct in our identification so that when we announce our protein result, we're accurate? And when we go, then step 3 is to get the protein done. We take it to GNS to measure the % nitrogen, and then we multiply it by some factor to get protein estimate. So this is Karen taking our samples. First she freeze dries them, then they weigh them in tiny little things. You can see how tiny it is at the end of the tweezers here. Right there is a little package of tinfoil with a little bit of pollen in it. It's about 5 milligrams. Yeah, whatever. Yeah, it's real tiny. Okay, and we put it in the machine. It's combusted. It's evaporated. Okay, and then that evaporation gives gas, and that gas gets passed through a thing called a mass spectrometer. The nitrogen and the carbon gas, and then that shows up on a screen, and then it gives some results, and Karen gives us the results. So that problem I told you about, this weed problem, that's really, really important. We do not want agricultural weeds, and we do not want environmental weeds. If we're going to do a Trees for Bees program and tell you guys, oh, go out and plant all these trees, some farmers are planting thousands and thousands and thousands of trees. Well, when you plant thousands and thousands of something that could become a weed, you're really pushing the limits there, where it's called propagule pressure. The more of it you have out there, the more likely it's going to run away and become a weed. So the first solutions would be, well, why not just use natives? Well, that's a good idea. Or just cultivate it. That's a good idea. And so we've got to take that to the farmers and see what they like. But the weed issue here, on the National Plant Pest Accord, which you know is absolutely illegal to propagate or use, we've got crack willow, which was the ones that the beekeepers think is great. We've got gray willow, we've got lantana and Scottish heather. They're all plants that you absolutely verboten, can't have them. The regional plant pest accords, all 10 regions, when we did our regional plant guides, all 10 regions are trying to get rid of gorse. And it's the linchpin of what beekeepers in many regions are using for keeping their bees going in the fall and into the early spring. Broom, same story. Blackberry, same story. Spanish heath, et cetera, et cetera, et cetera. So these plants, when I send out and talk to beekeepers, I say, I'm sorry, I can't do anything about those lists. You know, I mean, it's a political process, those lists of what's on there. But they are weeds, and they are in the environment, and they are hurting the environment in the regions that have listed them. So that's it, can't have them. And gorse and broom, it flowers twice. It's very high protein, 35%, a good one. Can't have hawthorn, that's on a list for some regions, 27% protein. Can't have sycamore, which was, we were delighted when we found the bees on this sycamore, but it's 26% protein, that's high. Anything over 20 is high, and can't have them. Because what that plant does is it disperses far into the native habitat, and it overtakes. So fine, okay. Cherry laurel, same thing. It's on one regional council, unfortunately. Well, it's only, I don't know, I was so sad to see it's on there. Can't have it. So let's just go native. Okay, well, I don't have a lot of natives done yet, but we have done flax. It's a winner. 32% protein, fantastic. Cabbage tree, 28, fantastic. Five finger, 20, but as Karen said, even if it's not high protein, you know, if you have it abundant enough, people in Taupo build up their beehives, beekeepers in Taupo, they have enough five finger to build up their beehives on five finger. So could that replace gorse? In some places, maybe. Pittosporum eugenioides, 25%. Don't anybody publish these results because I've got to get that confirmed, okay? So 25% for Pittosporum, really great. And another Pittosporum, I haven't identified it yet, it's 25%. So the natives are going to be a help, but when you talk to a farmer, they have some reason they want exotics, or actually, when we talked to ECAN, we went to ECAN, and they said, we do not want Hoharia populnea. We do not want Hoharia, you know what they are? They're lacebark. I don't want Hoharia sexticellosa. In fact, we'd rather you plant an exotic. And I said, you'd rather us, this is the nature, you'd rather us plant an exotic than to plant those two North Island Hoharias that are going to genetically swamp the South Island Hoharia. So you see we've got this eco-sourcing and the genetic swamping. That's what so we've got native weeds that we can't have. So you see why I have to do my research. You can't have almost everything we look at. Okay, so I came to Eastwoodhill because this is where the diversity is. 1,663 species out here, 5,000 cultivars, and et cetera, 3,500. And so Barry Foster was the one who told us to come here, and we did, and he's right. He put his bees on here. He got it would have been over 1 million bees. The 24 hives would be over, I think John calculated, it was over 1 million bees that we had going on in here. So here's what we got from Eastwoodhill. Weedy species. Yes, you can't have hawthorn, but what about if there's a cultivar or a species related that's as good as hawthorn, but it's not, okay, so what about this beautiful Crataegus fruscale, the cockspur hawthorn, 24% protein, and it's not on a weed list. I know. That's the problem, is that now you see part of the Trees for Bees in the background is to do the weed risk assessment. There are some cultivars where you can be pretty sure it's never going to be a weed. You can just look around in your garden and see what's spreading. The sycamores are always spreading, okay? And so we have that, and it's difficult, and you're right. And so I'm in between the ecologists and the beekeepers, with both of them yelling at me. But I think with careful work, I mean, that's why I don't want anyone to go out and say this, publish this yet, because we've got to put it through a weed. I'm going to show you have to put it through a weed filter as well. But maybe, maybe, I don't know. Portugal laurel, now the cherry laurel got on one region's list. I don't know how, most of the prunus are probably no. Unfortunately, they do spread a lot. But 25% protein, that's pretty good. And honeysuckle, it's unbelievable. Bees have feeding frenzies on honeysuckle, but honeysuckle's on the list, on a weed list, but is this species on a weed list? Does it behave like a weed? So we can study that problem. Will this become a weed? Okay? Don't know. Translucent honeysuckle, again, unbelievable source. Really good nectar. We see the bees taking nectar and pollen simultaneously from this plant. That's 19%. It's good enough. Now, so there, you're right. We have to make sure these things are not weeds, but are not going to become a weed. But you can look at those ornamentals and say, well, it's related, but it's reproductive. I am a reproductive biologist, by the way, a floral biologist. So you can look at it and say, well, we know that the way this thing is pollinated or dispersed, or the way it behaves, its gender system or its reproductive system, it's unlikely to become a bad weed. And so you can do that kind of research on plants. Now, one thing we found were things that were not on weed lists, the whole genus wasn't in many cases, and we got what we call common, and in fact, Danny Fraser came up with this. The curator was here at Eastwoodhill. He said common and undervalued and underutilized species. That is, these are plants that are going to do really well on a farm, but people just, I don't know, I just don't know that you can do it. Box elder, 21% protein. I don't know how weedy that is, but we have to do the research and find out. But I have never in my life seen a tree as loaded with pollen as that. This is all the anthers here. See the, where's my little thing here? See the, it's just dripping anthers. And there's the bees. And so that's amazing. Amur maple, 23% protein. Really good, all the maples, very good candidate species. Flowering ash, 21% protein, all the ash, very good species. Are they weeds? Don't know, have to look at that. But fantastic amounts of pollen on them. Now we can look at high maintenance species that fruit trees are well known to be really good, but the farmers say, I don't want to have apples and I don't want to have pears because, and they're really high protein, they don't want to be pruning and spraying. But you can have an ornamental, right? And you don't have to prune or spray it at all. I mean, you just stick this apple out there, and it's got the protein, 27%, 26%. Both of these are Eastwoodhill. Apples, fantastic. I got 4 more minutes to talk. Yeah, okay. And so apple, oh, and this apple, oh gosh, this is so beautiful. It's up by where Barry's hives are. And it's pink buds and white flowers, but I don't have a species name on it yet. 23% protein. Now, these are more uncommon species for New Zealand farms. Photinia, you see them on some farms around here, but a lot of people don't have it. Well, that's 25% protein. It's not a weed. Landcare, this is a shadbush, it's Canadian, and it's beautiful, 24% protein. That's down by the pond down there. Absolutely stunning, loaded with flowers, and the bees really love it. Ilex macrocarpa, my favorite, has beautiful perfume, 21% protein, and I don't think it's a weed. Beautiful tree, big tree. Mountain dogwood, 20% protein on this one. And a very interesting flower. Oops, oh wait. Very interesting flower structure. And then here's our prize, Staphylia bladdernut. I never heard of it. It's in the United States, and it's an edible nut. I couldn't believe it. The bees really love it. It was out there kind of early in September, and flowered for ages and ages, and had, what did I say it had? 32% protein. Well, I think, Karen, we got to check that one again. That's really, really, really high yeah. So we'll do that one again, but it might be 32%. And there's the nut that's not mature yet, but there's the nut that you can eat. Okay, now let's throw another, wrench in the, I mean, it's funny, you get really excited about a plant and then you find out, you can't have it. Well, so then I read a chapter on toxic plants. I found, now, a few species and some habits are toxic. In drought and cold conditions, a plant will be more toxic. The lack of forage of other things to dilute the poison will make a plant toxic. The nectar or the pollen, either one or both, can be toxic. It can be toxic to the bee. It can be put toxic stuff into the honey and be toxic to the human. It could be toxic to the cattle and not the sheep, or the sheep and not the cattle, et cetera, et cetera. So toxicity is an issue. So now we have to filter our database for toxicity. And we know that tutu is very toxic for honey. It kills humans, or really makes them sick. Caraca kills the bees, we know that, so you can't have that one. And Ko-Fi, a lot of beekeepers vote for it. And we even planted some on our demonstration farms, but the literature says sometimes some species in some conditions, under some weather conditions in some regions, it's toxic. So now our biggest prize was quercus, mainly because of the volume. Those are very, very big trees. There's the little person, that's me. I'm a very little person right there. And that's the tree just down on, down over the hill and here. And it's a Quercus robur, and we found a lot of oaks here. In fact, oaks are the one genus that has more species than anything else. And more accessions as well, I think. And those are all of them, and they range in their protein value from 18 to 22%, so we'd say that's pretty good. And you can't normally see them very well. They blend in with the, you can't even hardly, they just blend in with the flower. But I highly recommend this Mexican oak because in very few years, you've got, look at it, well, I'll show you closer. It's just dripping pollen, dripping anthers. But then I read my toxic chapter and it says, yeah, oaks are toxic. And I go, what do you mean oaks are toxic? How can it be? It's the core. It's like very important in England, oaks for bees. And it's on all the bee lists internationally. But one report from Russia said some bees got sick on them once, and it's not been repeated. So that's a question mark. I don't know if it's toxic or not. But I think there's a lot of oaks that are very suitable for farms, and I think they're a very good one. Now, this is a real embarrassment, because, is that the end? Oh, but I have the panel discussion a little bit of time, just a couple more minutes. Horse chestnut, we actually published in the Gisborne Herald that it was great for bees, and then I find it in the toxic. I should have read I didn't know about this chapter. It's a book, so. Yeah, horse chestnut, the bees, they go to it, and it is toxic. It really is. There's chemicals in it, not good, not good. So there you are. new to the international bee plant list is our gray eye, wonderful for probably nectar. Chinese cork tree, not so good, but the bees love it. And we've got 800 trees in our bee plant list. Lots of questions: the role of gorse, the role of willow and protein content, predicting noxious weeds, autumn plants for winter prep. I'm going to skip over this, but I'm just going to say here that we are going to go to Palmerston North for 6 months and study all the willows to bits, find out their protein, their flowering time, and make recommendations for the farmer, because you can expand the flowering time of oak by putting in a sequence of 5 species or so. And instead of getting 1 or 2 weeks of flowering, you're going to get a month or 2. I'll just skip through that. Here's our demonstration farms. That's a list of what we did plant. And 2 demonstration farms, 400 meter by 5 meter on 1 arable farm, and within 4 months we got good growth because of the way they herbicided it, so there's no competition. They used combi guards, so there's no browsing, and the combi guard protected against the herbicide, and they also, because it's Canterbury, it's irrigated. So that's 1 demonstration farm. Here's our other demonstration farm, 300 by 5 meter plot. Same thing, arable, permanent apiaries on these farms. And so we already have our shrubs flowering. Our California lilac, hebe, viburnum, and rosemary are all already flowering. The bees are already visiting, and we're only 6 months into our demo farm. So that's good. And Peter Hare, we have Peter Hare is here, and Pale Badger are working together on a sheep and beef farm. We have put in 370 plants, tree lucerne and viburnum, and I don't know if it's viburnum, it's tree lucerne, a prairie tree. Lots of good trees. And they're on, in Lake Rawa something, sorry? Sorry? Ripper, yeah, okay. And anyway, so the conclusion I want to make here, and there's lots of questions outstanding, but the conclusion I want to make is that for spring, I think our research in Eastwoodhill has shown that pollen dearth is an artifact on farms, which exists, berries taken me to farms, we've seen it, is an artifact of the low diversity on farms and public land. It doesn't need to be that way. Absolutely not. There's lots and lots and lots of species to plant. Can we solve it without weedy species? I think we can, but the challenge is sleeper weeds, which are weeds that they're not weeds until you put lots of them out there. So, yeah, but if we do good weed risk assessment, I think we can get through that filter too. So that's just a little bit about the future. We don't need to go into that. And I think that's the end of the talk. Oh, I had a sponsor page. Oh, yeah, right, sorry. That is not up to date. We've got new sponsors since, and I just want to say there's lots and lots of organizations and companies that believe in us and want us to get this research done for them.